Hello everyone, I'm Kishan K C from RIT. I'm presenting my work, Interpretable Sparse Encoding of Sequences for Protein-Protein Interaction Prediction. Proteins are the functional units within an organism that interact with each other to form molecular machine, and these molecular machines are responsible to carry out various biological and molecular processes within an organism. So it is important to study such interactions to understand biological phenomenon as well as to discover putative drug targets. Various computational methods have been proposed to identify interactions between proteins given their sequences. However, these models fail to provide interpretable outcomes and are computationally expensive during data preprocessing as well as model training phase. To address these challenges, we propose sequence encoder with sparse and structured regularization as shown in the figure below. Taking amino acid sequences as input, our model learns sequential and contextual information of amino acids from the sequences. Based on this contextual information, our model selectively focuses on a specific set of amino acids. Specifically, our model employs a sparse gating mechanism to learn a sparse mask on the amino acid sequences, thus promoting the interpretability. The model then uses this sparse representation to minimize the statistical distance between interacting proteins and maximize the distance between non-interacting proteins. We adopt mini-batch setting where we take a batch of protein sequences and encode them to their representation. Once we have the representation, we take all the positive and negative interactions between these protein sequences and optimize the loss function. This makes our model efficient. Based on our experiment and real-world data set for yeast and human, our model achieves better performance than state-of-the-art methods. We also observe that our method with a sparse gating mechanism improves the performance by selectively focusing on a small subset of sequences. Based on training time comparison, our model shows significant improvement in training time when the number of interaction increases. We interpret the sparsity mask learned by the model and find that for yeast, nearly 19% of amino acids are selected, and out of selected, 59% of them actually align with the motifs. We perform 3 sample case study where we choose sequences with motif on different regions of the sequences and found that our model's prediction highlighted by red align pretty well with the motif from PFAM library. I would like to thank my team members and the funding agency. Thanks for listening to my talk.